morning, everyone. As you trickle in, I'm just going to start presenting this morning's Grand Round speaker, Ben Liu. So Ben Liu is one of our graduates, and he actually exemplifies what we're trying to do in the Cancer Center in terms of building a pathway for training. So as some of you know, we have a whole list of T32s and K12s, and our goal is that people go from one program to another. And so Ben, who graduated from NYU Medical School, came here as a resident, and then joined our fellowship. And as a fellow, he joined Dr. Herbst's T32, and now he's a trainee on the K12 in immuno-oncology. The only problem is that Ben sometimes does things a little bit backwards. So he's currently finishing his PhD, but yet he's a faculty member, and this makes it extremely complicated when it comes to the paperwork of the K12. So if there are other trainees in the room who are hoping to come up through this pathway, please do it in the right order. But all that aside, okay, that's true too. So if you want to go by the chaotic method, then all I can say is I strongly recommend Dr. David Heffler as an amazing mentor. He's done a great job with Ben, who is doing amazing work on actually 3 major projects. One focuses on brain metastasis in lung cancer, one on melanoma and liquid biopsies, and that's the one he's going to be talking about today. And the third one is single-cell RNA sequencing studies of glioma patients treated with anti-TIGIT antibodies. So there are very few people who can shoulder all of this while being a chief fellow and a T32 trainee and a K12 trainee and everything else and a dad, I think, and everything else that Ben does. So without further ado, I'd like to welcome Ben to give us his presentation.
Thank you so much, Dr. Kluger, for that very kind introduction. Good morning, everyone. Hope everyone had a very nice Thanksgiving. I can tell you that I'm very thankful to be standing up here on the podium today and for the opportunity to share some of our work that has been supported by the Skin Spore over the past 5 years. I do have to say that I think it speaks a lot to our cancer center community that we're willing to amplify even junior investigators such as myself. And I'm incredibly grateful to my mentors, Dr. Heffler and Dr. Kluger, for nominating me to represent our team on this project. So I've titled my talk, Immune Liquid Biopsies, Remote Learning and Remote Control. And the topic here is a little bit different than the liquid biopsies that I think many of you are familiar with, which are more tumor-centric, and I'm specifically referring to circulating tumor cell-free DNA. But I think what we're starting to realize is that these liquid biopsies are really powerful companion diagnostics that are really trying to become game changers in care. And it's my hope that with additional work on immune profiling, that these two will start to emerge as important tools to help us improve our care for patients with cancer. And so I have no personal financial disclosures. Some data in the presentation was generated in collaboration with Repertoire Immune Medicines. And just to briefly go over the structure of my talk, I'm first going to talk a little bit about some evidence that we have that the broader systemic immune response is really a critical component to anti-tumor immunity. I'm then going to review some rationale and prior work that's been done in this space of immune profiling in the blood. And then I'm going to talk through 2 stories that we have, one which is published and one which is being prepared for submission, that really focuses on using the T-cell receptor as a molecular barcode to help us understand what is the relationship between T cells in the tumor and T cells within the blood. I'm going to close by discussing a little bit of our early efforts to try and translate our biological discoveries into clinically relevant biomarkers. And so just to start, as we all know, immune checkpoint inhibitors have really revolutionized the way that we treat patients with cancer. And it's in large part due to work such that's been done by Dr. Kluger and many of you out in the audience. And the gold standard for the potential that immunotherapies have for treating patients with cancer remains in melanoma, amongst other cancer types. But you can see that these are really practice-changing survival curves from the Checkmate 067 trial, which was a frontline trial looking at anti-PD-1 and or anti-CTLA-4 for patients with advanced melanoma. But you can also tell from these curves that about 50% of patients still fail to derive long-term benefit. And I think it calls into question why is that? What are the mechanisms that are causing them to not be able to mount a systemic immune response that can result in tumor rejection? And are there markers that we can use to try and identify these patients? And are there therapeutic avenues that we can explore by learning with this information? And so a better understanding of the fundamental determinants dictating clinical response are really needed. Just to review what our current understanding of immune checkpoint inhibitors are, when immune checkpoint inhibitors were first introduced into clinic now over a decade ago, the thought was really that these agents target negative signals within the local tumor microenvironment and thereby reinvigorate T cells, which we believe to be the primary effector immune cells and resulting in tumor rejection, reinvigorating these local T cells to recognize the tumor. Well, we've since come to learn, though, that at least in part, the potential for immune checkpoint inhibitors to mount successful tumor rejection is the need to induce immune responses beyond the local microenvironment. And several groups, including those here at Yale, have identified the tumor-draining lymph node, for example, as one reservoir for tumor-specific stem-like T cells that help to regenerate and sustain anti-tumor immune responses. This is nicely illustrated in preclinical models whereby we can block lymphocyte trafficking. And in doing so, we see that anti-tumor immunity is really impaired, and the efficacy of immune checkpoint inhibitors is also limited. This has been demonstrated by several groups, including two papers out of groups from Yale, from Nick Joshi's lab, and then also from Marcus Bosenberg and Richard Flavel's lab. We also know that immune checkpoint inhibitors not only recruit new T cells to the local tumor microenvironment, but that these T cells may have actually be recognizing different antigens. And we're assessing that based off of their T cell receptor sequences, termed novel clonotypes here on the right. And perhaps some of the most exciting data that's emerging is the potential benefit of immune checkpoint inhibitors to work in early stage disease, even after the tumor has been removed, the macroscopic tumor has been removed. And so these are disease-free survival curves from recent trials that have explored anti-PD-1 therapy in the adjuvant and neoadjuvant settings. And what these data reinforce is that checkpoint blockade really potentiates immune surveillance beyond the local microenvironment and helps to prevent tumor regrowth and disease recurrence. So in this setting, we really believe that a systemic immune response is an important contributor to effective anti-tumor immunity. And our underlying hypothesis for this project was that blood-based tumor-related T cells really have distinct characteristics and can be informative of local tumor immune microenvironment. Our translational goal is therefore to try and identify clinically relevant biomarkers, which can be obtained non-invasively through the blood to try and assess and inform us on anti-tumor immune responses. And so prior work in this arena have nominated several blood-based biomarkers. However, uptake into the clinic is likely challenged in part due to the lack of specificity. So several serum cytokines, which we know to be context dependent, are not widely used, or due to inavailability of certain techniques within our clinical labs, such as the ability to determine T cell receptor diversity or clone sizes in clinical labs. And so our general approach has been to first take a deep dive and deep look into T cells within the tumor microenvironment. And in order to do that, we employed using single cell sequencing. This is a technique that allows us to simultaneously characterize both the gene expression profile of individual cells, and in the case of T cells, also the full length T cell receptor sequence. Now, the T cell receptor is really an essential component to everything that a T cell can do. The T cell receptor is what allows T cells to become activated when it encounters its cognate antigen. And the global diversity of the T cell repertoire is really, really immense. And so having a high resolution view of the sequence is really important. When T cells do encounter their cognate peptides or their antigens, they become activated and they proliferate. And all of these sister clones share the same T cell receptor sequence. And so in that sense, the T cell receptor sequence is really a useful molecular biomarker for us to be able to link T cells that are clonally related within the tumor and the blood. And we can then ask the question based off of its gene expression profile, how are these cells changing and what can we learn in these two spaces? And so in this first portion of the talk, I'm going to talk a little bit more about using TCR as a molecular barcode. And I'd really like to just acknowledge Liliana Luca, who was a former postdoc in our lab and junior faculty member in our lab, who's now an independent investigator in France. She was really an important architect in driving this project forward, this initial story. And so for this initial, the initial look at using TCR as a barcode, we performed single-cell RNA sequencing and T-cell receptor sequencing from in blood and tumor from 11 patients with stage 4 melanoma. These patients all had mixed histologies and treatment histories. And the purpose of this initial look was to try and assess a global look at what these clonally related T cells, global features of these clonally related T cells. The way that we went about identifying tumor T cells, which we think are relevant to the anti-tumor immune response, was by looking at how clonally expanded they are. This helps us differentiate T cells that we may be located within the tumor, but that are not actively participating in the anti-tumor response. We then linked these over into the blood, and we termed for this initial story, these cells, these clonally related but blood-based cells as circulating tumor infiltrating lymphocytes, which I'll refer to as circulating TILs. And so these circulating TILs are a relatively rare population in the blood. They are comprised of less than 10% of our total T cells. And you can see that they're predominantly located within the CD8 T cell compartments. So what I'm showing is on the right is a dimensionality reduction plot of our single cell RNA sequencing. The circulating TILs are highlighted in dark green. You can see that they're predominantly distributed within the CD8 T cell compartment. These cells are clonally expanded not only within the tumor, but also within the blood. And that interestingly, this population seems to accumulate over the course of your disease. And so we can perform differential expression analysis to try and take an unbiased look at the transcriptional features of this population. These circulating TILs are the ones that are located in the right, and all other blood T cells are located on the left. And we're focusing on CD8 T cells in this case. What we find is that they share features of high cytotoxicity, tissue residence, cell migration, tissue homing, and importantly, as kind of a check, they lack features of naive or memory markers such as CCR7, TCF7, and these are features this is important to us because it reinforces the fact that these are cells that have been activated and are actively participating in the immune response. We can also ask the question, how are these circulating TILs related to the features of tumor cells or tumor T cells? And so the first analysis that we did was we generated a gene set that is characteristic of expanded T cells within the tumor. We then took a look at the expression of this expanded TIL gene set within our circulating TIL population as compared with all other blood cells. And we do see that there is an enrichment for this population, or this gene set. We can also ask, are circulating TILs characteristic of gene sets of T cells which are specifically expanded within the tumor, and thereby removing genes that may be just generally associated with clonal expansion? And we again see that there is an enrichment for this gene set. One thing that I will point out is that there are several hallmark genes which have been described as important features for T cell dysfunction or tumor exhaustion, such as our co-inhibitory checkpoints, such as CTLA4, TIM3, which is encoded by the gene HAVCR2, and then the transcription factor tox. PD1 is also found, although not listed, displayed here on this screen. And so we can see that circulating TILs are not representative of features of exhaustion within the tumor, but that there is a good concordance between a cytotoxicity signature between this population and the degree of cytotoxicity within the tumor. And we can also ask the question, are T cells that have been described to be predictive of response to immune checkpoint blockade, are those T cells also found within the circulation? So this is work out of Nir Hacohen's group, whereby he generated 2 gene signatures of CD8 T cells, one that was enriched in patients who responded to immune checkpoint blockade, another which was enriched in those who were resistant. And then we took a look to see whether or not these gene signatures, what are the global distribution of these cells. What we find is that the resistance signature is really only enriched within T cells, which are exclusively found within the tumor and not found within circulation, whereby the response signature is found in T cells that are shared in both compartments. And I think what this point illustrates is that a key component to a good response to immune checkpoint blockade is prior systemic priming of the anti-tumor immune response. And so just to summarize from this first portion of the talk, we've described that circulating TILs are enriched with genes and that are associated with clonal expansion specifically within the tumor, and that the degree of cytotoxicity, but not exhaustion, are reflected in circulating TILs. We find that tumor T cells that are predictive of immunotherapy response are also shared within the blood, and that hallmark features of a productive anti-tumor immune response may be reflected in the blood. So one of the assumptions from this early work was that the most tumor relevant or most relevant T cells to the anti-tumor immune response are those that are most largely clonally expanded. And around the time that we were performing this initial work, there were also groups that had described that you can use strictly the transcriptional signature of T cells to accurately predict whether or not these T cells were neoantigen specific, whether they're truly tumor specific. One such paper was out of Steve Rosenberg's group at the National Cancer Institute, whereby he described 2 gene signatures, one for CD4 T cells and one for CD8 T cells, that can, with high accuracy, predict whether or not a given T cell was likely to be neoantigen specific. And so we simply asked the question, can the transcriptional identification of tumor-specific T cells improve our understanding of the blood and tumor relationship? And so we then applied this transcriptional prediction to our own data. And so these are the CD8 T cells that I had shown in the previous section. And this is all from the tumor. And we've identified those that we think are likely tumor neoantigen specific. I apologize about the coloring of the graph on the right over here. But what we can see is that using our previous definition of expanded or unexpanded T cells, that you have to trust me on the coloring, but the vast majority of them are also predicted to be neoantigen specific. But I think an important point is that of the unexpanded, there's also a portion that we were not capturing before and that are actually unexpanded within the tumor microenvironment. To functionally confirm that these predicted T cells do in fact recognize neoantigens, we collaborated with Repertoire Immune Medicines and with data that was generated by Ruth Halban's lab as well to analyze whole exome sequencing and bulk RNA sequencing to be able to predict for each individual patient neoantigens and tumor-associated antigens. We then ran these peptides that were synthesized in a relatively high throughput manner against select T cell receptor sequences. and tested for reactivity. What we find in this data is that the vast majority of those neo-TCR predicted T cells account for basically all of the T cell receptor sequences that elicited functional reactivity, and that the only clonotype that was reactive but was not predicted to be neoantigen specific was in fact reactive to CMV, and this peptide was included as a negative control for by the repertoire team. And so using this approach, we then analyze cutaneous melanoma, 17 patients with cutaneous melanoma who are immunotherapy naive. And we chose to focus on a more biologically homogeneous cohort to try and really eliminate any treatment-related effects. We then apply the neo-TCR8 and neo-TCR4 signatures to predict and identify reactive T cells, and in a similar fashion, link them back into T cells within the blood based off of their T cell receptor sequences. In total, we identified about 7,000 reactive CD8 T cells, which again reinforces that this is a relatively rare population. We again find that they're predominantly CD8, that they're highly expanded and also have a restricted clonal diversity. So what I'm showing here on the right is a linearized metric for the degree of clonal expansion within the blood. And the tumor match reactive are the ones that are predicted to be reactive based off of their tumor transcriptional signature. Unreactive are ones that were unreactive, but also found within the tumor. And then also the ones that were only found in the blood. We also find that there's a higher frequency of previously reported tumor antigen-specific TCR sequences in our reactive population. And to do this analysis, we use publicly available databases of TCR sequences that had been annotated with their functional epitopes. To try and understand in a more specific manner the transcriptional features of these reactive T cell population, we collaborated with Dr. Eval Kluger's group, and they had developed a novel computational method for identifying the differential abundance of certain populations. Wes Lewis was a graduate student in his lab who applied this to our data set. And what we find is that we can identify a subpopulation of cells that are differentially enriched for tumor reactive T cells. A look at the differential expression gene signature shows that in the unmatched and unreactive cells, there's again an enrichment for naive and memory markers in line with our previous work. There's a high degree of cytotoxicity that's both found within our reactive and our unreactive populations. There's signs of cell trafficking, tissue residence, and NK-associated markers. And there's one marker in particular that really stood out to us, and this is the killer cell immunoglobulin-like receptor family, which KIR2DL3 is one of those subtypes here. And the reason why this is interesting to us is because this work, KIR-expressing CD8 T cells, was recently described in autoimmunity and in infection as being an important mechanism for restoring peripheral tolerance. So just a little bit about the KIR receptor. So they're best understood for their function and role within NK cells, and they impart a negative suppression signal upon encounter with class 1, class 2 MHC. So they're in fact co-inhibitory signaling within NK cells. Within CD8 T cells, though, they denote this regulatory-like T cell, which is analogous to the Li49 expressing CD8 T cells that Harvey Kanter's group had described in mice. But these cells have a high expression of the transcription factor helios. And although the mechanism isn't fully understood, they can target pathogenic T cells in autoimmunity and infection and kill them in a contact-dependent manner. And so in essence, this is an alternative mechanism to try and eliminate hyperinflamed or hyperactive T cells. Their role in tumor immunity is not really well described or well understood. So turning back to our data, we can take a look at gene signatures that are characteristic of these KIR-CD8 regulatory cells in autoimmunity and compare them with our population, our reactive population in melanoma. And what we find is that there's a broad expression of the KIR family of receptors. There's also high expression of cytotoxicity and NK-associated genes, in addition to many of the other features that I pointed out before. Importantly, there's a high expression of the transcription factor Helios, which is thought to be essential to their regulatory function or their suppressive function, and a notable absence of co-stimulatory molecules. You can also see from this slide that this gene signature seems to be fairly specific for this reactive subpopulation of reactive cells as compared with all other CD8 T cells found within the blood. We can also perform gene set enrichment analysis, and we do find a statistically significant enrichment for the top 200 genes of human KIR-CD8 T cells found in autoimmunity. And we can also ask the question, does this population, or does this KIR-CD8 T cell population, does it represent a distinct differentiation state, or is it part of a continuum within clonally related T cells? And I performed pseudo-time trajectory analysis here, which attempts to try and order biologically related cells along a continuum. And what we find is that there seems to be a branch trajectory here, and that in unsupervised analysis, we also find that Helios, which is encoded by the gene IKZF2, also came up as one of the most differentially expressed and associated genes along the trajectory. We can also ask the question, so if we think that these are regulatory cells within the blood, do they maintain their transcriptional state within the tumor? And in essence, we're trying to understand what might be their role within the tumor microenvironment. And so what we can do is we can trace these cells based off of their T cell receptor sequences back into the tumor and look at the transcriptional profile. And what we see is that this KIR-CD8 T cell transcriptional profile is largely maintained within these sister clones within the tumor microenvironment. We have ongoing work in collaboration with Dr. Marcello D'Estasio and the Department of Pathology to try and better characterize these histologically using spatial multi-omic analyses in the tumor microenvironment. And so just to conclude from this second portion of the talk, we've demonstrated that transcriptional signatures can identify a subset of tumor reactive T cells which are not clonally expanded. Differential abundance techniques can help us identify a subpopulation of these reactive T cells, which largely resemble KIR-CD8 regulatory T cells, and that these KIR-CD8 T cells seem to represent a distinct differentiation state, which is preserved in the tumor microenvironment. And so an important question for us is, what is the clinical relevance of this T cell population? And because we can't perform single cell sequencing on all of our patients, we really wanted to move towards markers that could be assessed within the clinical laboratory. I'm specifically referring to using flow cytometry. And so what we wanted to do is to move from transcriptional features over to protein level cell surface features, which are conventionally used in flow cytometry. And in order to do so, we collaborated with Dr. Steve Ma's group and Yuanxin Chen and Jiping Wang, our postdocs and graduate students in his lab, who primarily worked on this project. And we asked them to see whether or not they can construct a classifier that is limited to clinical variables and also genes that are specifically associated with protein, cell surface proteins. We also restricted ourselves to genes which are known to correlate both at the transcriptional level and also the protein level. And so they constructed a lasso logistic regression model, which can accurately predict or classify cells as being likely within our subpopulation or not. And when we then apply this back into our single cell data set, what we find is that this tumor reactive KIR-CD8 T cell population seems to be associated with the poorer survival. And what we did here was we simply split our cohort into a high expressing group and a low expressing group using a median cut point. And what I can tell you is that it doesn't matter whether or not these patients were immunotherapy naive or immunotherapy resistant. The mere presence of these cells seem to be associated with worse overall survival. Moving towards applying our classifier, applying these features to flow cytometry, we then asked them whether they can construct a hierarchy of these genes and protein markers in order for us to be able to develop combinations of markers that we can assess on flow cytometry. And so they first use single cell data that we had generated, but this includes protein level expression from site-seq data. And what I'm showing you here is that the expression of KLRD1, as an example, that the site-seq expression is relatively similar to what we would see on flow cytometry. We then constructed a decision tree model, which allows us to assign a hierarchy and summarizes a combination of markers. And using this model and this combination of markers, we can accurately classify cells 91% of the time. And with the caveat that this is still ongoing work and that we have short interval follow-up for this exploratory cohort, we do see an early trend in separation curves that is in line with what we were seeing before. That is, those who have a higher proportion of this subpopulation seem to have worse clinical outcome. And to validate both our transcriptional data and also our protein level data, we established a collaboration with Dr. Benjamin Fairfax at the University of Oxford. He's a melanoma oncologist who has generated bulk RNA sequencing data and also flow cytometry data from over 200 patients with melanoma prior to treatment and also on treatment. And so we're looking forward to seeing those results. And so just to summarize, we believe that the induction of systemic immunity is really a critical component to successful anti-tumor immune responses, but that clinical biomarkers, which allow us to profile this population, remains an unmet need. We use single-cell technologies to try and provide insights into the relationship between T cells within the tumor and those within the blood. And that we've identified a subpopulation of tumor-reactive KIR-CD8 regulatory T cells, which may actually suppress anti-tumor immunity and negatively correlate with clinical outcome. I think this is largely exploratory, but potentially, if we can identify this cell population within a clinical cohort, we may be able to explore a new therapeutic avenue for targeting these cells. And so with that, I'd like to just take a moment to acknowledge all the people who've made this work possible. I think first and foremost, we need to acknowledge the patients and families who are very generous in donating their tissue and blood. But also, I'd like to thank them and acknowledge them just for the motivation that they provide all of us for the work that we do in the clinic and the lab. I'd also like to thank my mentors, Dr. Hafler and Dr. Kluger, for their unending support and really the opportunity to perform this research. In addition to members of the Hafler lab, so Liliana Luca, I had mentioned before, is an independent investigator in France. Pierre-Paul and Nick were also essential and instrumental in generating data on the study. Our collaborators both internally here at Yale and also externally. So Dr. Yval Kluger's group and Wes Lewis for their work on the differential abundance analyses, and Steve Ma, Yuanxing Chen, Jiping Wang for their work on our constructing biomarker classifiers. As I mentioned before, Marcello D'Estasio, we have an ongoing collaboration to explore the spatial orientation of the cell population. And Dr. Benjamin Fairfax is a collaborator who's going to help us explore this population in a larger cohort. I'd also like to acknowledge our collaborators at Repertoire Immune Medicines, in addition to the Yale Skin Spore, whose support has really made this effort feasible, and also to the core facilities here at Yale. And a personal thank you to both David Braun and David Schoenfeld, who unfortunately couldn't be here in person today, but they were incredibly generous in help with their thoughts and also with their time in helping me prepare for this presentation. And also for my funding sources, the T32 and the K12, as Harry had mentioned before. With that, I'd be happy to take any questions.
Thank you, Ben, for a terrific talk. Any questions? So while people...
So we have a few online.
Oh, yeah, let's do that.
Yeah, so I don't know if people want to unmute, but I see that Sri Vatam. Has there been an effort to isolate and phenotypically characterize these CUR CD8 T cells? I'm curious to understand the uncommon state of CD8 T cells. So yes, there has been work in other contexts to do that. And I didn't show the data today, but we have also done that in melanoma and have largely validated the protein level immunophenotypes of these cells. The next question is from Marcus Bosenberg. He asks, do you have a hypothesis as to how CUR CD8 regulatory T cells negatively affect anti-cancer immune responses and outcome? You know, I think the mechanism for these CUR CD8 T cells is still really not fully understood. The hypothesis, as kind of demonstrated here on this slide, or our hypothesis, is that they're somehow impacting tumor antigen-specific CD8 T cells in the tumor microenvironment. I'm currently in the process of setting up assays to try and assess this functionally. But my guess would be, and it's also possible that they're impacting CD4 T cells, which is a more direct link from the autoimmunity literature. But we're first going to explore the CD8 component because of this negative impact that we see in tumors. And then the last question was, are these cells called regulatory based on their transcriptional features? So this cell population was, as I mentioned, described both in mice and also in human autoimmunity infection because they are able to actually functionally kill autoreactive T cells. And so it's not simply just based off of transcriptional features, although our data is certainly inferring from the transcriptional expression.
I actually have a follow-up question to Marcus's. Do you think that these positive CD8 T cells might stick and you can revert them to the negative phenotype, even because they occur? Because there are antibodies that are being actually given to humans that do that.
Yeah, so I think the NKG2DA antibodies, which impact the kind of analogous co-stimulatory molecule, not the co-inhibitory molecule, have been tried, and I don't think the data has been all that great for it. But in terms of the plasticity of this cell type, I think, especially based off of our the trajectory analysis, I think that it is interesting to try and explore how plastic the cell population is. It does seem like there's a branch differentiation trajectory, but we just don't understand quite yet how these cells are really being generated, under what context, and to really truly demonstrate their function.
Just speak up, yeah.
So with this model in mind, when you look at cohorts that are receiving IO, there's a relationship between poor survival and CUR positive cells strengthen compared to if you look at cohorts that aren't receiving IO?
Yeah, so the p-value remains about the same, actually, in both cohorts. And I think that's a really interesting point, but whether there's a subpopulation of patients where this is a primary immune secondary resistance mechanism, I think is worth exploring.
Wonderful talk. These KIR suppressor cells that Mark Davis identified, a really hot issue in human immunology now. But just looking at the slide again, Do you think the tumor reactive T cells may express a ligand for TIGIT CD155? Have we looked at that?
Are you talking about the regulatory cell, the cure cells, or the ones on the left?
Well, the regulatory cells that care positive express TIGIT.
Yes, they do.
The ligand is CD155, and what we're learning about what CD155 engagement does to cancer cells as per PPG, I'm just wondering if CD155 is expressed on the tumor reactive T cells. Have we looked at that?
I mean, I'm inferring, we haven't directly looked at that, but I'm inferring and I'd be guessing that the expression, if present, is very low. But I think it's definitely worth exploring. I think the effects of anti-TIGIT and anti-PD-1 on this cell population, for example, is something that we can take a look at.
Absolutely.
Another question, have you done similar analyses in other tumor types? In your lung cancer study, do you see the same new cell population of CD8 Treg?
So I haven't, so for those who aren't able to hear online, Dr. Kluwer asked whether I've also found this cell population in other tumor types. I haven't specifically looked under this lens. You know, one of the reasons why I think maybe if this population is real, it may not be as well described is because it has a large transcriptional overlap with other cytotoxic CD8 T cell populations. And so we really need to look carefully the enrichment. Cures, for example, can be expressed on just activated CD8 T cells. And so we really need to look a little bit more carefully at some of the other markers like Ikaros or Helios. There is one more question online. So from Ying's side, why cure CD8 T cell product? Can you prevent cure CD8 T cell product? I mean, I apologize. I'm not sure I fully understand. I'm not sure if you mean a T cell product that's targeting cure CD8 T cells. I'm not sure if you're available to unmute and discuss. But I do think that exploring a therapy that would target or impact these cells would be of interest.
Okay, thank you, everyone. Thank you.